be talking about my master's project, a little bit about what I've done in the last 2 years. Can you guys hear me okay? Thumbs up, thumbs down? Cool. So I'll be talking about resolving relationships in Leucaena through de novo transcriptomics. So before I get into the methods and what I actually did, I'd like to talk about the background of Leucaena, since most North Americans have never actually heard about the genus. It's in the Fabaceae family, mimosoid clade. There are 24 species, 19 of which are diploid, and 5 are tetraploid, allotetraploid specifically. Most are trees, but there are some small shrubs. And the native range is, the northernmost range is Texas and southern New Mexico, and it extends down into Peru, with most of the diversity in central and southern Mexico. So it's got the typical mimosoid morphology with the globose or subglobose inflorescences, the bipinnately compound leaves, and the leaf-associated extrafloral nectaries. They can be found on the rachis or the petiole. Those are associated with ant relationships, hermaphroditic relationships. And actually, I've got some pods here. These are the typical pods you'll find on species. They vary in size and shape, but this is what they look like. And come back to me after the session. You can taste test one of these if you'd like. So it has a long history of use in Mesoamerica, where it's used as a protein supplement. But the common name is guaje, and it's actually the origin of the city and state name Oaxaca in Mexico. It means home of Leucaena, Huaxin. And archaeological remains suggest that it's been in use for at least 5,000 years. And it still, to this day, has cultural significance among Mexicans for food source, primarily as a protein supplement, as I said, but also as medicine and timber. And one of the more esoteric uses of it is a delousing agent for chickens. I thought that was a little bit interesting. And there are 2 sites here on the left. This is Gila-Nikitz. You might have heard of this name. It's the first, the Leucaena was discovered in the same strata as the first known domesticated corn archaeological site, or maize, sorry. And on the right, you've got Monte Alban. It's a 2,000-year-old site. In the foreground, there's a Leucaena tree as well as an agave plant. This represents the typical cultivated plants you might have seen at the time, about 2,000 years ago. And here's some people processing Leucaena. You've got the unripe seeds being pulled from the pods and collected for consumption. And here they are in markets. You've got Leucaena esculenta on the right, Leucaena leucocephala on the left. Those are 2 popular species that are used in food. So global usage of Leucaena has, usage of Leucaena has expanded to a global range, exclusively in the tropics, but it's extended to Africa, Southeast Asia, Australia, primarily as an agroforestry crop. So it's used as cattle fodder, soil amendment, biofuels, and oftentimes in alley cropping. And as a result of this human translocation, Leucaena leucocephala is recently listed as one of the 100 worst invasive species. And here it is in the agroforestry setting. You've got alley cropping on the top left, where there's maize interbedded with Leucaena. And on the right, there are some Leucaena that are in preparation for intercropping with banana plantations. And on the bottom 3 panels, you've got what are called valley bulls. They're popular cattle in Indonesia. Here they're fed on an exclusively Leucaena diet, 100% Leucaena diet. And I'd like to switch gears from the background of Leucaena to the background of phylogenetic analyses done on Leucaena. In the 1990s, that's when the molecular work began, focusing on chloroplast loci, very few loci. But then the most recent and most comprehensive study we have is from 2011 by Govindaraj et al. They've got a few more genes from the chloroplast region and also picked up some nuclear genes. But still too few genes, and the genes were not informative enough. And these red points on the phylogeny represent weak or no support nodes. And you can see that clades 2 and 3, the relationship there is not well supported. The placement of cuspidata up here is not well supported, and a lot of the relationships within clade 1 are not well supported. So we intend to develop more well-resolved diploid phylogeny using phylotranscriptomics. RNA-seq data has already been developed for a multitude of, or a variety of purposes. It's a multi-purpose data set. Two of the analyses I'm interested in working on are the sequence-based phylogenetics, which I'll be talking about today, and the SNP detection for high development or high throughput species markers. I've already done that, but I'm not going to really talk about that today. So the materials we used with that RNA-seq data, we assembled transcriptomes for 18 of the 19 diploid species, and those are de novo transcriptomes. There's a 19th diploid species that we lacked seq material for, and we were unable to get RNA-seq data for. So instead, we got genomic DNA and had a partially constructed genomic DNA. We also have outgroup transcriptomes from 3 other mimisoid species. And the approach we decided to use was the Yang and Smith approach. I'm sure many of you are probably familiar with this approach. It was successfully demonstrated on the order Caryophyllales in 2015, and we adopted that protocol. In the 2015 version that was published to Yang Ying's Bitbucket site, the phylogenomic data construction set tutorial, she included a new step that used well-annotated proteomes as sort of references for beta homology search. So transcriptome data was aligned to these proteomes. Sequences of transcriptomes that aligned well were clustered together in homolog clusters, and then orthology assessment was conducted to get a set of orthologs for phylogenetic inference. So we carried out the protocol as designed until we got to the point where we could make a choice, and that was specifically the orthology assessment stage. There are 4 methods that we could have chosen. We chose the 1-to-1 ortholog approach. And this approach, after masking monophyletic tips in the gene trees, we selected only gene trees that had 1 representative per tip on the gene tree, exactly 1 representative. So this method retained fewer loci, but it reduced the risk of including paralogs in the ortholog clusters. So as I mentioned before, we only had genomic DNA for Leucaena puebllana. So we had to take some additional steps that weren't part of the original Yang and Smith approach. And here's an outline of what we had to do. So we had our assembled Leucaena puebllana genomic DNA, and we transcribed and translated the long course from that genomic DNA, and then reduced redundancy so we had unique sequences. And then we piped those back into the unrefined homolog clusters from the previous analysis that excluded Leucaena puebllana, the strictly transcriptome analysis. And so ultimately we ended up with 505 ortholog clusters with roughly 750,000 aligned bases. And using those aligned bases and those ortholog clusters, we used 2 tree building methods to generate our species tree. The first was the concatenated gene tree, specifically RAXML, and the second was the coalescent-based method, which was Astral. And we've got jackknife support values on this tree here. If you look above the branch, that's the Astral support value. If you look below the branch, that's the jackknife support value, or the RAXML support value. And some general ideas you can get from this tree is that the 3 clades that were previously established are retained. And the relationships within these clades that were not well resolved before are well resolved for the first time. There's a couple notable exceptions. The RAXML tree building approach had weak support at 2 nodes. Specifically, the placement of Leucaena had 54%, and then the placement of cuspidata had no support. But these green dots represent places where the previous phylogeny failed to resolve well, and we were able to recover good resolution. And then we started to look at the concordance and conflict within these gene trees. The important parts of these pie charts are the red and green portions. Those represent conflicting percentages of the gene tree. And you'll note that within clade 1, there's a high degree of conflict. And as we've seen in other talks today, that's not uncommon, and it actually provides some clues as to the evolutionary history of our tree here. And actually throughout the tree there's quite a bit of conflict, but particularly in clade 1 there's a lot of conflict. So what does that tell us? The evolutionary history of the genes is not necessarily correlated with the evolutionary history of the taxa themselves. And likely incomplete lineage sorting and/or hybridization are the cause of this conflict. So previous studies have shown that these diploids readily hybridize, but distribution data shows that they're allopatric in distribution. All diploid species are separated from one another. So do they actually have a history of hybridizing? We don't think so. And then if we look at branch lengths, there are very short branch lengths, specifically within clade 1. And I'll point out where all the short branch lengths are. The red dots signify nodes that have less or greater than 50% conflict. And these nodes actually correspond to areas of the phylogeny that have historically been difficult to resolve. So what does the tree reveal about Leucaena? The conflict and short branch lengths are consistent with incomplete lineage sorting as a result of rapid speciation. Also, elements of the phylogeny are consistent with morphology that's been observed before. So for the first time, the leaf morphology, specifically the large leaflet clade, is shown to be monophyletic. This clade is denoted by its leaflets that are wider than 1 centimeter, and their leaflet bases are mostly symmetric, only slightly asymmetric. The remainder of the taxa have very narrow leaflets, narrower than 1 centimeter, and their bases are more or less asymmetric. Additionally, there's an interesting bark morphology. Most of the species in Leucaena have a rough, corky, brownish colored bark, but this clade too has described as galvanized zinc texture. It's very metallic, and it's a very thin bark. So our results largely are consistent with the three clades. Well, they are consistent with the 3 clades that have been previously described. And I haven't talked about it today, but the geography is supported of these. The geography of these 3 clades is supported by our phylogeny as well. There's a high likelihood of rapid speciation resulting in complete lineage sorting, particularly in clade 1. And then we've got some novel phylogenetic results. So relationships within clade 1 are resolved for the first time. A large leaflet clade is shown to be monophyletic for the first time. And also the placement of cuspidata and the relationship between clades 2 and 3 are supported for the first time. So now that we have this robust phylogeny, we've got some interesting questions to ask and have answered. So these allotetraploid hybrids, we know that they're very recent hybridization events, likely due to human translocation. And now we'd like to explore the origins of these 5 hybrids and then develop ecological niche modeling so that we can look at how the niche of the allotetraploid offspring compares to the niche of the diploid parentage. And I'd like to thank most of my lab is here today. I'd like to thank them and also my committee and past lab members and additional support, some people here as well. So with that, I'll take any questions.
These toxic?
Okay, so Leucaena has mimosine in it, which is a non-canonical amino acid, but there are low concentrations of it. Our whole lab is eating them, and we're alive 2 weeks later. So if you want to take a risk, you can try them.
Thank you. Any other questions? We can take one more question.
What genetic frequency did you use?
So we used 63% gene resampling.